Microbial structure. They are as unrelated to human beings as living things can be, but bacteria are essential to human life and life on planet Earth. Although they are notorious for their role in causing human diseases from tooth decay to the black plug, there are beneficial species that are essential to good health. Prokaryotic cell structure. For example, one species that lives symbiotically in the large intestine manufactures vitamin K, an essential blood clotting factor. Other species are beneficial indirectly. Bacteria give yogurt its tangy flavor and sourdough bread its sour taste. They make it possible for ruminant animals like cows, sheep, and goats to digest plant cellulose, and for some plants, soybean, peas, and alfalfa, to convert nitrogen to a more usable form. Bacteria are prokaryotes lacking well-defined nuclei and membrane-bounded organelles, and with chromosomes composed of a single closed DNA circle. They come in many shapes and sizes, from minute spheres, cylinders, and spiral threads to flagellated rods and filamentous chains. They are found practically everywhere on Earth and live in some of the most unusual and seemingly inhospitable places. Evidence shows that bacteria were in existence as long as 3.5 billion years ago, making them one of the most oldest living organisms on Earth. Even older than the bacteria are the archaeans, also called archaeobacteria, a tiny prokaryotic organisms that live only in extreme environments, boiling water, super salty pools, sulfur-spewing volcanic vents, acidic water, and dipped in the Antarctic ice. Many scientists now believe that the archaea and bacteria developed separately from a common ancestor nearly 4 billion years ago. Millions of years later, the ancestors of today's eukaryotes split off from the archaea. Despite the superficial resemblance to bacteria biochemically and genetically, the archaea are as different from bacteria as bacteria are from humans. In the late 1600s, Anton van Leeuwenhoek became the first to study bacteria under the microscope. During the 19th century, the French scientist Louis Pasteur and the German physician Robert Koch demonstrate the role of bacteria as pathogens or causing disease. The 20th century saw numerous advances in bacteriology, indicating the diversity, ancient lineage, and general importance. Most notably, a number of scientists around the world made contributions to the field of microbial ecology, showing that bacteria were essential to food webs and for the overall health of Earth ecosystem. The discovery that some bacteria produced compounds lethal to other bacteria led to the development of antibiotics, which revolutionized the field of medicine. Microbial structure in both unicellular, multicellular, and acicellular. Capsule. Some species of bacteria have a third protective covering, a capsule made up of polysaccharides or complex carbohydrates. Capsules play a number of roles, but the most important are to keep the bacterium from drying out and to protect it from phagocytosis by larger microorganisms. The capsule is a major virulence factor in the major disease-causing bacteria, such as Escherichia coli and Streptococcus pneumoniae. Non-encapsulated mutants of these organisms are avirulent. They don't cause disease. Cell envelope. The cell envelope is made up of 2 to 3 layers: the interior cytoplasmic membrane, the cell wall, and in some species of bacteria, an outer capsule. Cell wall. Each bacterium is enclosed by a rigid cell wall composed of peptidoglycan, a protein sugar of polysaccharide molecule. The wall gives the cell its shape and surrounds the cytoplasmic membrane, protecting it from the environment. It also helps the anchor appendages like the pili and flagella, which originate in the cytoplasm membrane and protrude through the wall to the outside. The strength of the wall is responsible for keeping the cell from bursting when there are larger difference in osmotic pressure between the cytoplasm and the environment. Cytoplasm. The cytoplasm or protoplasm of bacterial cells is where the functions for cell growth, metabolism, and reproduction are carried out. It is a gel-like matrix composed of water, enzymes, nutrients, waste, and gases, and contains cell structure such as ribosomes, a chromosome, and plasmids. The cell envelope encases the cytoplasm and all its components. Unlike the eukaryotic, bacteria do not have a membrane-enclosed nucleus. The chromosome, a single, continuous strand of DNA, is localized but not contained in a region of a cell called the nucleoid. All the other cellular components are scattered throughout the cytoplasm. One of the most components, plasmids, are small extrachromosomal genetic structures carried by many strains of bacteria. Like the chromosome, plasmids are made of a circular piece of DNA. Unlike the chromosome, they are not involved in reproduction. Only the chromosome has the genetic instructions for initiating and carrying out cell division, or binary fission, the primary means of reproduction in bacteria. Plasmids replicate independently of the chromosome and, while not essential for survival, appear to give bacteria a selective advantage. Cytoplasmic membrane. A layer of phospholipids and proteins, called the cytoplasmic membrane, encloses the interior of the bacterium, regulating the flow of materials in and out of the cell. This is a structural trait bacteria share with all other living cells, a barrier that allows them to selectively interact with their environment. Membranes are highly organized and asymmetric, having two sides, each side with a different surface and different functions. Membranes are also dynamic, constantly adapting to different conditions. Flagella. Flagella, singular flagellum, are hair-like structures that provide a means of locomotion for those bacteria that have them. They can be found at either or both ends of a bacterium or all over its surface. The flagella beat in a propeller-like motion to help the bacteria move toward nutrients away from toxic chemicals, or in the case of photosynthetic cyanobacteria, toward the light. Nucleoid. The nucleoid is a region of cytoplasm where the chromosomal DNA is located. It is not a membrane-bound nucleus, but simply an area of the cytoplasm where the strands of DNA are found. Most bacteria have a single circular chromosome that is responsible for replication, although a few species do have two or more. Smaller circular auxiliary DNA strands, called plasmids, are also found in the cytoplasm. Pili. Many species of bacteria have pili, singular pilus, small hair-like projections emerging from the outside cell surface. These outgrowths assist the bacteria in attaching the other cells and surfaces, such as teeth, intestines, and rocks. Without pili, many disease-causing bacteria lose their ability to infect because they are unable to attach the host tissue. Specialized pili are used for conjugation, during which two bacteria exchange fragments of plasmid DNA. Ribosomes. Ribosomes are microscopic factories found in all cells, including bacteria. They translate the genetic code from the molecular language of nucleic acid to that of amino acids, the building blocks of proteins. Proteins are the molecule that perform all the function of cells and living organisms. Bacterial ribosomes are similar to those of eukaryotes, but are smaller and have a slightly different composition and molecular structure. Bacterial ribosomes are never bound to other organelles as they sometimes are in eukaryotes, but are freestanding structures distributed through the cytoplasm. There are sufficient differences between bacterial ribosomes and eukaryotic ribosomes that some antibiotics will inhibit the functioning of bacterial ribosomes, but not our eukaryotes, thus killing bacteria but not the eukaryotic organisms they are infecting.